professor, physician, epidemiologist. I am Dr. Sri Banerjee. Hello, I'm Dr. Sri Banerjee, epidemiologist, biostatistician, faculty member. And in this lecture, I'll be talking about a concept that is oftentimes confusing and difficult to grasp, and that is the concept of epigenetics. Traditionally speaking, when we use the word genetics, there's a lot of connotations that arise from this term. For instance, for scientists, of course, this may mean genetic counseling for individuals that may have certain genetic abnormalities like Down syndrome. For people that are studying genetics, this may mean understanding Mendelian genetics, studying how natural selection takes place. If you're a virologist, this may mean that you're studying antigenic drift, antigenic shift. So the word genetics has different connotations. Listen, we have 23 pairs of chromosomes, and so 22 autosomal chromosomes, ones that codify most of our body, and then one pair of sex chromosomes. And if you're a guy, then the pair of sex chromosomes is XY, and if you're female, then it's XX. So that's the genetics. And until about 30 years, that was the extent of a genetics lecture. That was it. But now we have found, now that the whole genome has been revealed for everyone to kind of do research on and do more discoveries on, the Human Genome Project has finally been revealed in the last decade and a half. So now we're paying more attention to something called epigenetics. And these are the modifications, the modifications that take place once the genes have been implemented. So once the genes then are transcribed and translated, during or after that process, there's several types of modifications. So that's what this paper is all about. So before I even got a chance to talk about this paper, we had to have this 3-minute discussion. So now that we're talking about epigenetics, this is actually a very important article connecting epigenetics to social determinants of health. And why are we even talking about social determinants of health in connection to epigenetics? Because traditionally thinking about genetics are non-modifiable characteristics. But as it turns out, because I told you epigenetics is actually the modifications that are made after the genes have been codified, this means that the characteristics could then be inherited into future generations. So genes have a way of interacting with social conditions. And that is the premise of my talk today. That is exactly what I'm saying. Currently, there's a lot of funding that is being allocated towards this. There's about 25 million, actually, to be exact, $26 million allocated very recently to the NIH, specifically for social epigenomics research. So if you're somebody looking to specialize into something that's not quite genetics, you don't feel comfortable with the genetics portion, then social epigenomics can be a good field to try to put all of these aspects together. So, of course, we already know if you're a public health practitioner, you know the social determinants like the back of your hand. It's the conditions, it's the conditions in the environment in which people born, live, work, play, worship, all the conditions. Those are what the social determinants of health are. Now, of course, this is taking place throughout the life course. Now, this becomes important when we're considering epigenomics, that these are the conditions, the social determinants are the conditions in which we live, in which we are born, live, work, play, worship, and but this is going on throughout the life course. What does that mean? That means that the genetic conditions and the epigenetic expression of the genetic conditions are changing over time. So now we're looking into specific examples that have been used for this. For instance, in disadvantaged neighborhoods, there tends to be a higher availability. I've studied this firsthand. There's higher availability of alcohol, tobacco, fast food restaurants, and combined with unhealthy food options, lower availability of food options. So, what does that make for? If you have a lot of food deserts and more alcohol, tobacco places, then these are areas of deficiency. These are areas where people will naturally yield poor health outcomes. And that is a fact. So this needs to be really addressed. And so when we're combining some of these social issues of increased exposure to chronic stress due to lower availability of, say, healthy food options, then of course a lot of these get closely intertwined with genetic level processes, biological processes, and behavioral processes. So then that combines, and there could be free radicals, oxidative stress, all of these things, maybe from polyunsaturated fats, and then so these in turn result in epigenetic alterations. So you can read more about some of these processes of how this happens, of how neighborhood level effects start to impact stress hormones, and then that then impacts epigenetic alterations. So there's a nice figure that has been provided here. One specific epigenetic factor that the authors talk about is the severity of asthma. So, in fact, epigenetic studies show that epigenetic alterations actually impact the severity of asthma. So more of this untranslated 5' untranslated methylation takes place, the more the severity of the asthma level. And so this is, of course, research on this specifically then is an example of where social determinants have been combined with epigenetics. And that's because asthma is a social determinant condition. So when we're looking for the immigrant paradox, for example, um, and of course, if for those of you that may not have heard about this, it turns out Hispanic immigrants have better health outcomes as compared to non-Hispanic whites, especially when they first arrive in the country. But then as time goes, then this this health advantage actually diminishes. So this could be due to socioeconomic gradient that there is, and sometimes a lot of these socioeconomic gradients and social determinants can actually play a role in epigenetic modifications affecting molecular processes. So, social determinants have direct consequences on molecular processes, and that is the premise behind the idea of epigenetics. So one major way that this takes place is through DNA methylation. Um, so when you're studying chronic diseases, if you're looking for specific ways that there could, for instance, asthma, the connection between the asthma and then the DNA methylation there. If you're looking for connections, epigenetic connections is one way um, that a risk factor versus outcome can be demonstrated. So another DNA methylation, this is an example of a good one. Um, and so the DNA methyltransferases, um, they actually donate the S-adenosine methionine group and to complex sites. And so this causes differential methylation as well, and differential expression of different genes. Um, so for those of you who may not know, actually, for the DNA methylation, the four common base pairs in DNA is cytosine, guanine, adenosine, and thymine, CGAT. So these four base pairs comprise all of genetic code. And so, as it turns out, the 5-methylcytosine, so this C, 5C, is where the methylation is taking place, and that's the most common type of methylation, DNA methylation, that takes place in order for epigenetic modifications to take place. So these are a lot of times known as kind of the CPG site, because the C has been modified. So there's other sites where this sort of methylation can take place, and actually, if you go back to some of these modifications that take place, epigenetics takes place in many different places, more places than you would at first think. For instance, parental imprinting is one area where epigenetic modifications take place, where how if a lot of your father's genes are expressed more so, than epigenetic oppression of some of your mother's genes actually don't allow expression, and then vice versa can be true as well. And sometimes there's actually a mixture, and this is not just true for the human species. This is seen in fungi and other organisms and species as well. Another point I would like to make before we move on too far down this is that epigenetics is a mitotically inheritable modification to DNA that do not directly alter the sequence. So they can be just regular or through genetic imprinting, as I described to what that was. So the mechanisms responsible for regulating these modifications, they're transcriptional, they can be post-transcriptional, translational, and post-translational. Okay, so we have gone through the first type of modification. I'm going to quickly go through the rest of them. The histone modifications, those are important. And I've described them elsewhere, but you can read about them here as well. Histones are very tightly packed proteins, so that a lot of genetic information can get tightly packed. And here also is a good place for some epigenetic alterations to take place. And so sometimes they act as erasers even. So then you have microRNA and long non-coding RNA, which can also play a role in epigenetic modification. So those are the main parts. But the intersection, I think, is the most important, and this is how all of these can kind of come together to provide the effect. So social determinants of health on the left, of course, this is important, but epigenetic modification, the histone modifications, DNA methylation, microRNA expression, all of these epigenetic modifications, they can have an impact. So over the lifespan, early in life, fetal alcohol syndrome, or fetal alcohol spectrum disorder, as it is known now, developmental disorders, and asthma. Within midlife, you can have addiction and substance abuse, metabolic syndrome and cancer as a difficulty, and then in late life, Alzheimer's. And all of these are impacted by race, ethnicity, and many other sociodemographic factors that really need special consideration and special understanding. So I think this is a good stopping point. You can go through the rest of the paper and look through the details of it. There's a lot of information packed in there, so if we were to go through all of it, then we wouldn't be able to talk about everything, but chronic stress is one that is emphasized in this when we're talking about chronic diseases and epigenetics. Metabolic syndrome is another big topic that is talked about, connecting chronic diseases and epigenetics. And then some of the physiological changes that happen late in life, and any type of cancer that might be taking place, cognitive decline like Alzheimer's disease, and then the limitations and current challenges that are around for social epigenomic studies. There's a lot of changes that are taking place during the individual's lifetime, a lot of epigenetic changes, and to be able to capture that snapshot, how we're going to do that, that is quite a bit of a challenge as well. And so teasing out some of the gene environment interactions can really require some collaborative research. So the authors actually talk about an interesting statistical approach for elucidating causal inference, and that is the inverse probability of treatment weighting. So this is a propensity weighting for inclusion in statistical analysis used for controlling for selection bias in epigenetic studies. So if you do a lot of epigenetic studies, that's something good to be thinking about. But this sums up some of the epigenomic updates that have been taking place currently, and students really, or researchers, or those that are looking into this topic should really look to understand this as another epidemiological chronic disease framework that can provide you with further insight into understanding complex topics. Thank you for listening.